Okay, hello and welcome to the exit seminar portion of, did I get on the screen?
No, the camera's on.
Welcome to the exit seminar portion of the PhD defense of Michaela Woods. Just to orient everyone to the process, first I will do a short introduction, then you will hear the exit seminar for Michaela, which is an overview of her PhD research. Then we will have time for anyone in the room to ask questions. Following that, the committee will convene for the defense portion of the process, which is focused not only on the oral presentation, but also on the dissertation itself, which she has submitted for review. Following the defense portion, we will gather outside here in the atrium. I'm going to estimate an hour or so after the conclusion of the oral presentation, either to celebrate or to drown our collective sorrows. I want to seriously thank everyone for being here and for all you've done to make the day possible. Granting a PhD is the highest award we make in academia. It's the ultimate achievement and literally places the person in extremely rare company. The best data I could find was that less than 1% of people in the world have a PhD, which Michaela may after this day. So this is quite an honor and an important moment, and we want to thank the committee, especially Dr. Bauer for coming up, the department and the university for supporting the process. Okay, so impermanence is a fundamental challenge in life, impermanence, and it's a foundational challenge for leading a research lab at a university. All the folks who join the lab are, in the end, just passing through. And in fact, it's our job to accelerate that process and support the process. It's core to what we do. Even so, there are times when it's very hard to navigate, and I'm here to tell you that Michaela Woods graduating and leaving my lab is going to be very hard to mitigate. You see, she has been an absolutely amazing graduate student here at the University of Dayton and served as a fulcrum for transformation in the lab and the foundation for the science that we've done over the last 4 years. Michaela has led a reorientation of the overall research mission of the lab to focus on ecological restoration. She picked up threads of projects and turned them into scientific products, and then launched new products and then wove all of that together into a new program, which you're going to hear about today. She was incredibly productive in the laboratory, and in fact, practically all of her dissertation has already been published. She has 4 chapters already published that were submitted to the committee as reprints from journals. A 5th chapter that is basically ready to go or gone, about to be submitted, very close, and a 6th chapter, which you're going to hear about today, which is a huge new project at the Great Miami Mitigation Bank. That project established a foundation for a successful grant that I wrote, that Michaela was a co-author on the grant, to the Green Acres Foundation, and has set the stage for 2 new master's thesis projects, which are just now getting off the ground. In fact, this 6th chapter has set the stage for what may be a decade or more of new work in the lab. I want to point out that Michaela managed all this work while having a baby through a global pandemic, and now about to have another baby. So this is quite an amazing accomplishment. I don't want to hurt anyone's feelings, but I do want to say that in my view, Michaela's probably been one of the most productive scientists at the University of Dayton over the last 3 years, not graduate students, not in the Department of Biology, overall, if you rank order to everyone, UDRI, engineering, she's been one of the most productive scientists at the university. Importantly, on many of those papers that she's published, there have been co-authors, including undergraduates, and also some of our collaborators from the 5 Rivers Metro Parks have been co-authors, indicating a truly community co-created research program that she's been involved with. Beyond this, she's also spent time on several side projects, including peer-reviewed journal articles, and most recently generated a publication in the journal Science that included a journal cover. There's not very many people at UD that's ever been true for. It's a rare feat. To recognize all of these efforts in 2021, Michaela was awarded the John J. Comer Graduate Award for Excellence in Research in Ecology. I hasten to add that Michaela has also been an award-winning teacher in the biology department. She has specifically been an innovator in developing curriculum around the use of the R programming language for code-based analysis of biological data. In fact, Michaela has had a hugely positive influence in training students in my lab, students in other labs, and in our curriculum in general. She developed an R module for introductory biology labs, meaning that every single student who comes to our program now has some access to coding, which they did not have before she was here. She has also done a lot of training on more advanced topics, including being an instructor of record for an advanced statistics class in the department, which is also a rare feat. One interesting contribution Michaela has made in the realm of teaching is that early on in her program, she got interested in online teaching. And in fact, she took a graduate level course at the UD Teaching and Learning Center on teaching online. At the time, she was told by a faculty member, "Why are you wasting your time taking that? We don't teach online." And then a matter of days later, we heard a thing called COVID-19. Not long after, and the entire curriculum was online. And so that's hashtag bad advice. I don't know if you use hashtags, but because Michaela had this training, she helped develop an online version of our introductory biology lab, which then became a model for that lab when everything went online. So she helped support that. To recognize all these efforts, Michaela was awarded the Gerald Willis Award for Teaching Introductory Laboratories in 2020, and also was the inaugural winner of the Biology Innovation Award, which was also in the year 2020. I don't know what that was for, maybe the R coding or the online innovator in biology. And the award was invented basically because of her efforts, I would say. In summary, it has been an unbelievable honor and privilege to have Michaela as a PhD student in the McEwen Lab. She has helped set an amazing standard and been an irreplaceable member of our team. So I bet you can tell why I'm lamenting impermanence. Alas, we must move forward and just suffer the loss, but not yet. We have one more hour left, starting right now. So help me welcome Michaela.
Hey, thank you, Ryan. So I'll go ahead and get started on my dissertation talk called Restoring Degraded and Invaded Landscapes, a Soil-Based Approach. First, I want to make sure my slides move. There we go. First, I want to say thank you to all of the people that have helped along the way. First, thank you so much to Dr. McEwen for allowing me so much freedom and flexibility in my program. I've been able to do pretty much everything that I wanted to do in the last 4 years, and I don't feel like that's super common for a lot of people, so I'm really grateful. Thanks to my committee members, Dr. Chelsea Prather, Jonathan Bauer, Albert Burkini Bonstein, for all of the awesome conversations we've had and for all of the advice along the way. Daniel Clark, Saren Frankenberg, Grace Atia, Kate Lloyd, Emily Jones, Melanie Muratori, Molly Simonis, and Erin Anya Frack have been some of my closest friends and supporters throughout the last 4 years. I couldn't have done this without them, always there for me when I'm complaining all the time. So thank you. And then thanks to my funders, the Ohio Invasive Plants Council, Ohio Biological Survey, the University of Dayton, Green Acres Foundation, and Five Rivers Metro Parks. A special thanks to Grace Deach, Meredith Cobb, and Lisa Zaromsky, who have been really influential for our work at the Five Rivers Metro Parks. All of my studies take place there. They've been really, really helpful in making sure that we can do great science and have access to all of the land. The McEwen Lab is a huge place. There's lots of people that come in and out and lots of people that I worked with throughout the years. So thank you to everyone who has helped me, and I really appreciate you. So with that, we'll go ahead and get into the talk. So I want to start my talk framing it around global farmland abandonment, because throughout the world, we're seeing that farmlands are being left fallow. They're no longer being used for farms, and they're being repurposed, and different land uses are happening here. So anywhere in the world that's not white in this map is where farmlands are being abandoned, so where land use change is able to occur. This is really exciting in the time of the United Nations Decade on Ecosystem Restoration. So the United Nations has declared this decade of 2021 to 2030 to be focused on ecosystem restoration, taking lands that had served a purpose that had been anthropogenically influenced and reverting them back to some sort of native habitat. The goals for the UN are to restore 30% of priority ecosystems, to prevent 70% of species extinctions, and absorb 50% of the carbon emitted since the 1700s. So this idea of farmland being abandoned throughout the world presents a really exciting and unique place for all of this restoration to occur. We have land that can be used. Zooming in to where we are in the Midwest, US, we have this issue when we think about our abandoned farmland, which is a field of exposed dirt. Basically, there's a land that's been constantly cultivated. There's nothing growing here. This is just kind of left like this when farmlands are left fallow. So yes, so this is a common thing that we see within the Midwest. This presents a really unique opportunity to restore forests specifically. In the 1600s, most of the eastern United States was covered in forested land. And then by the early 1900s, we see that a lot of our forests were no longer there. They've been clear cut for things like agriculture, urbanization, or logging purposes. So these old fields could present a really unique opportunity to restore forested ecosystems throughout the eastern US. Thinking about what we would want our forests to look like in Ohio specifically, here I have a map of pre-European settlement vegetation. So if we're thinking about a native ecosystem, we want to go before European settlement to see what was here originally. In this region in Montgomery County, Ohio, where all of my studies take place, we typically see either beech forests or mixed oak or oak sugar maple forests. So in my dissertation, I really focused on oak forest restoration. This is in part because oak regeneration is really complicated, and oaks are globally important species, and they have a declining overstory dominance throughout the world. So oaks are typically limited by things like dispersal. They spread through acorns, which are typically carried by wildlife. But if you don't have oak trees nearby your restoration sites, you're probably not going to get your acorns into that site, so dispersal might not be happening. They're really, really picky about where they grow. They've got specific microclimates, habitats, and soil nutrients that they need to do well. So if they're not planted into an ideal environment, they may not do very good, especially once we get to the competition, because oaks are super slow growers. So anything that can grow faster than them and establish near them is probably going to outcompete those oak species. In fact, in one study we did in chapter 1 of my dissertation, published in Forests, with undergraduate Katie Hickel and our Five Rivers Metro Park collaborator, Meredith Cobb, we planted 60 oak trees in a forest restoration, went back and looked at them after 7 years, and out of our 60 trees, only 1 survived. So this means just going out and planting oak trees into sites, into these restoration sites, might not be enough to actually generate an oak canopy. So it begs the question, how do we regenerate these oaks that are really, really important? And that kind of leads me into this question or idea of, if we've got all of this abandoned agricultural land, and this is what it looks like, but we want oak maple forests, how do we start this transformation? How do we make this happen? Or how does it happen on its own? And does it happen on its own? So if we dive into some of the differences we see between these habitats, our fallow agricultural field has no trees, herbaceous layer, and it doesn't have shelter or food for wildlife. Whereas our oak maple forests have trees of various ages and species, a very biodiverse herbaceous layer, and ample habitat for wildlife. If we go below the surface and think about what's going on in the soil of these ecosystems, our fallow agricultural field has been experiencing really intense industrial agriculture for the past couple of decades, up to hundreds of years, where we've got heavy machinery plowing through, taking off the surface soil and removing all of the plant material. So it's constantly being cultivated. There's nothing is living there. No plants are living there, I should say. And this also means that there hasn't been a chance for organic matter to accumulate on the surface of the soil. So it's very bare mineral soil that is exposed. Because there's constantly tractors and other equipment running over the soil, it's very compact, and there's not very much pore sizes or room for water to permeate through the soil. Altogether, this leads to a really limited niche space and biodiversity within these ecosystems. In our oak maple forest, instead, we see that there is a litter accumulation. So all of the plants or the trees in our forest are dropping their leaves, they're decomposing, and it's created a really nice organic layer of material on top of the mineral and clay soils. Because of the rooting behavior of the plants in these forests, there's also more pores available within the soil, it's aerated, there's water permeability, and there's more niche space for diversity. So going from a fallow agricultural field to an older forest, we might expect something like, or we might expect this succession to occur over the next 100 or so years, generating this forest. So this would be what happens on its own without intervention. Typically, what you would expect is that you go from bare ground to acquiring some weedy or early colonizing species, developing an herbaceous layer with some woody colonizers present, before our early successional tree species begin to form a canopy, which then shifts to later successional tree species like an oak hickory forest. So after 100 years, you would have trees of various sizes in the ideal canopy cover. And during this, we may also expect passive soil restoration to occur. So we would expect that as time goes on, we would go from a low water retention, low organic material, and depleted carbon and nitrogen within these soils to high amounts of water retention, organic material, and sequestered carbon and nitrogen from the plant behavior and from the lack of, or from the cessation of agriculture. So I wanted to understand, is this what we're actually seeing? Is this what's occurring in these forest restorations in the area in Montgomery County? So we created a chronosequence of restoration age to look at the soils from a 0, 7, 10, 50, and 100-year-old forest restoration. This is going to give us an idea of what's going on in all of these different steps of succession. So this study was published in Restoration Ecology. It's chapter 5 of my dissertation. I'm sorry, it's a little out of order to my committee. And it was published with then graduate student Sarah Frankenberg, undergraduate Jojo Adwakis, and our wonderful lab tech Kate Lloyd, as well as our Five Rivers Metro Park collaborator Meredith Cobb. The first thing I wanted to understand is how soil chemistry changed through time. So I wanted to look at the differences between soil chemistry at each of those time points. I did this using a principal component analysis. And the way that you read a principal component analysis is that things that are close together are really similar, things that are far apart are dissimilar. So I hypothesized that the soil chemistry associated with young restorations would be the most different from soil chemistry associated with old restorations. So I put them as far apart as they could go on my PCA. Next, we can map on our soil chemistry. So I hypothesized that these younger restorations would be associated with phosphorus and pH. Our older restorations would be associated with carbon, nitrogen, and moisture, and maybe something like sulfur wouldn't correspond to age, which is why it's kind of out there on its own. Then we can plot on the different ages of these forests. So my hypothesis was that through time, we would see a stepwise change in the soil chemistry from 0 years old here in this pink, all the way to 100, but that the 0 and 100-year-old forests would be the most different from each other. So they're going to be the farthest away from each other. Now, this is what we found, which is a lot less pretty to look at. But if you're generous with me, I do think that we see that stepwise pattern, which I'll show you. So our youngest restoration, that 0-year-old fallow field here in the peach, is the farthest away from that 100-year-old here in the purple. They do share some overlap, but it's okay. We see that the 7 and 10-year circles here are totally overlapped, which means they're really, really similar in that chemistry. And then if you're really generous with me, this blue 50 looks like it falls out in the middle of all of it, right? So we do see a stepwise change in the chemistry here. The younger forest restorations had more phosphorus, iron, aluminum, and more basic soils, whereas our older restorations had more moisture, carbon, and nitrogen. Now, what does that mean for tree growth? So we then took these soils into the greenhouse and planted Quercus macrocarpa tree seedlings for 6 months to see if their growth was impacted by these differences in soil. So we're looking at tree seedling mass here on the y-axis and restoration age on the x. And we see that the tree seedling mass was lower in soils collected from 0 and 7-year-old restorations than it was in the 50 and 100-year-old restorations. So not only are we seeing that soils are changing through time, but those changes are lending to an enhanced tree growth after 50 years. When we look a little bit closer at the chemistry and how it relates to seedling size, with seedling on mass on the Y here, soil moisture on the X for this graph, we see that with increasing soil moisture, we had larger trees. We had larger trees with decreasing pH. Our older restorations had a lower pH. And then increasing tree size with carbon, nitrate, and ammonia. So these changes to soil are happening through restoration, and they are enhancing tree growth. So I'm confident, or I'm okay with saying that during this process, soil restoration is occurring in a way that promotes the growth of target tree species. However, that didn't happen until 50 to 100 years after the restoration was initiated. So my next question then is, what can we do to speed up this process? Do we really need to wait 50 to 100 years for this to happen, or can we intervene and help establish a forest before that? So I have a 4-step plan that I've implemented to try to accelerate this process and see how we can improve the site for tree species. My plan consists of planting an intermediate prairie, using mulch to improve soil chemistry, using a soil transplant to enhance biodiversity, and then introducing tree seedlings. So step 1, if we can establish an intermediate prairie on that site, it provides a valuable habitat for native wildlife that has high biodiversity. So we're getting ahead of, we're getting to a native biodiversity faster. Additionally, if we plant target native species into the site before other species can establish, we could see a decreased likelihood of invasion because the space is already taken up by native plants. We use this as a means to test diversity of plant communities and how they resist invasion. So maybe if we have plant mixes with higher species richness, then there'll be less invasion because there'll be more niche space taken up by more plant species. We also wanted to look at legume concentration. So legumes are plants that take nitrogen from the atmosphere and make it into a bioavailable form within their soil. So if we have more legumes, there should be more nitrogen within the soil available for other plant species. So could this help with our tree establishment? And finally, prairie plants will really facilitate the restoration of soils by aerating the soils through all of their root systems, decreasing the bulk density, and by adding carbon directly to the soil. Our second step was to directly change soil chemistry with mulching. So we got locally sourced leaf mulch and added it straight onto our field site to add labile carbon and organic matter to the surface soil. This provides an insulation layer that can act as a temperature buffer as well for the soil. It can increase water retention, and some studies have found that it doesn't facilitate tree growth necessarily, but it can help facilitate prairie species establishment, which could be really important for our intermediate prairie community. And I would be really remiss if I didn't talk about soil fauna when we're thinking about restoring a forest. So our forests are creating and generating lots of organic matter that's being broken down by things like bacteria, fungi, nematodes, going all the way up the food web to our insects and wildlife. So they are really important in establishing a forested ecosystem. Specifically, I'm interested in mycorrhizal fungi, which are a really unique mutualist with plants, where they establish a relationship on the roots of plants, and they can provide water and nutrients to the plant in exchange for carbohydrates and photosynthetic materials. So I was really hopeful to get some mycorrhizae into our site, which is why we did step 3, so whole soil application to introduce native soil fauna. We went out into older restored prairies that were at least 30 years old and collected rhizosphere soil from those prairies and brought it over to our field site and sprinkled it onto the target restoration as a way to bring the soil fauna to our site. Finally, we brought the trees to the site as well to overcome dispersal limitation. So we planted oak trees directly within our site to see how they would benefit or not from these treatments. So my goal overall was to test the effectiveness of this plan. If we use these restoration techniques at the beginning of the restoration, will we see an enhanced outcome compared to just planting trees within a site? So this was funded by Green Acres Foundation and the 5 Rivers Metro Parks. We planted 4 prairie mixes, a high diversity forb and grass mixture that had 31 species and was 20% legume content by weight. The same high diversity mix, but with only 5% legumes by weight. A low diversity grass and forb mixture that was 19 species, subset from the high diversity with either 20% or 5% legume content by weight. Within each of these plots, we divided them into 4 subplots, one with seed mix alone, one with whole soil, one with mulch, and one with a combination of whole soil and mulch. So we can see how the seed mixes by themselves are changing the outcomes of the restoration, as well as how the soil amendments together and by themselves are changing the restoration outcomes. So we implemented the seed mix and the soil amendments in 2020. And between 2020 and 2022, we've been looking at prairie communities and the soil nutrient and microorganism activity. And then in 2022, we finally went out and planted trees. So we planted 480 trees into the site, and we measured their growth at the time of planting and after one growing season. So I have a couple preliminary results. In my dissertation, this is kind of a proposal at this point because there's so much data that it's just a lot to sort through, and we're still collecting more. So I didn't want to try to create a story out of that yet. But here are a couple of things that we're seeing. First, looking at tree growth rate over the 6-month time span from May to September of this year. We have Quercus macrocarpa growth here on the y-axis, and then combinations of all seed mix and soil amendments here on the x, all the way from that high diversity 20% legume with a control, meaning no soil amendment, to a low diversity 5% legume with the whole soil and leaf compost. So every combination that there was. And we did not see any changes in tree growth rate by treatments yet. This is not that surprising. This was one growing season. The trees were planted in their acorns with some acorns still attached. So they're getting a lot of their nutrients from the acorn still. But I'm really excited to see how this will change through time. We did see some shifts in the plant community through time. So you read this NMDS similar to that PCA, things that are close together, similar, things far apart are dissimilar. And what I want you to notice is that 2020 here in the pink is a little bit separated out from 2021 here in green. And then in 2022, we see our community pulling in another direction. So there are shifts through time. Specifically, what we're seeing is a higher species richness in 2022, but also more abundant woody and invasive plants in 2022. So the things that are enhancing our species richness in general may also be enhancing the establishment and survivorship of woody and invasive plants that maybe aren't our target. There was less bare ground in 2022, which gives us the idea that a lot of the niche space is being taken up. Establishment of our prairie community has happened. We're starting to see a lot more of our seed mix coming out now in year 3 than we had been in the previous years. So that's really exciting. And finally, we did also see a change in soil chemistry with time. So our 2022 soil samples are here in blue, and 2019 are in pink. In 2019, we saw higher exchange capacity, phosphorus, ammonia, sulfur, calcium, potassium, manganese, copper, and aluminum. That makes sense if you think about fertilizer use. So if this field had been fertilized up until the year that we started measuring the nutrients, it had things like phosphorus, ammonia, calcium, and potassium added to it every single year. So it makes sense that when you stop adding those things, they're not there anymore. What's really exciting to me is that we're already seeing a change in our soil pH, the amount of nitrate and the amount of organic matter, which means our plants are doing things, they're decomposing, they're there. The soils are changing in beneficial ways. So that's just a glimpse of what was going on. It's not a very clear story at the moment, but some considerations are that one growing season is not a very long time for measuring oak tree growth and success. So I'm really excited to see how that changes in the coming years. And we did see statistically significant changes in soil chemistry and plant community by the treatments that we use. So I just showed you through time, but we saw differences by the soil treatments and the plant communities. And that suggests to me that if those are enough to change our plant communities and our soil chemistry, they may be enough to promote tree growth as well. So stay tuned for those results. The future steps on this project's kind of being handed off to master's students Valerie Thurston and Madeline Gregory. We're measuring the fungal communities within the soil to see if that whole soil transplant was able to bring over beneficial microorganisms. Madeline is working on quantifying mutualist associations on the tree roots. So she collected some tree seedlings from the site and is looking for arbuscular and ectomycorrhizae on their roots. And Valerie's working on a really fine scale plant richness assessment, so we can see exactly which plants are in the plots and what they're doing. I did a pretty coarse assessment, so this is going to be a really exciting place to take the project. Okay, so I hope that from all of that, one thing you take away is that ecosystem restoration faces a lot of challenges. It's not straightforward. We're trying a lot of things to get it to happen and to get these trees to grow, but there's no clear answer. Now, this is made even more fun and exciting by what I like to call the pear predicament. So what happens when you don't get a fallow field and instead you go out to your field site and it looks like this? It's dominated by something else already. And here I'm going to talk about and focus on Callery pear. So what happens when old fields are dominated by invasive species? What if they establish in one of these earlier time frames? Do they grow concurrently with the other things that would usually grow? Do they take over the overstory? Do they become the dominant species? We don't really have the answers for that. We're not sure what's going to happen if you just let them go. Will we get an old growth forest or some sort of weird novel ecosystem? Who knows? So how do we restore land that's already occupied by invasive species like Callery pear? Callery pear especially loves all of these sites that are ripe for restoration. It escaped cultivation and spread into fallow fields, roadsides, and edges. Any disturbed environment, it's just like so excited to be there, which is so fun. Typically, when you want to get rid of your invasive species, you would cut, mow, burn, use herbicide. Once they're gone, you're like, great, this was successful. Unfortunately, some invasive species also change the soil where they've been growing. So not only you can get rid of them, but they can still impact the ecosystem in different ways. So Callery pear is unique in that it also just doesn't really mind being removed. It's like, try your best. This is a picture of a tree that was burned. And then you can see all of the dead stems on the tree. But Callery pear was like, I don't care, and just grew around it. Like, whatever, no big deal. Burn me again. I like it, it seems. Which is true. Like, we have seen that Callery pear does actually sprout more if it's burned, which is very great. And then it also will respond to herbicide with sprouts if you don't apply your herbicide precisely. If you use your herbicide precisely, you may see that you can get rid of it, but it is challenging. So this is a bit concerning because the ecology of Callery pear is relatively unknown. We don't know how it affects native species. We don't know how it's changing the soil. We just don't really know what it's doing when it's there. And just to give you an idea of Callery pear invasion and how it's changing through time, all of the green counties here are where Callery pear is present in 2015. Now in 2020, let's see, we've got a lot of the Midwest and a little bit of the East, but in 2022, we see most of the Midwest, and then a lot of the East Coast is now experiencing Callery pear invasion. If we look at the range expansion or the predicted future range, everywhere in orange here is a habitable place for Callery pear to survive. And so if it spreads into these areas, it's very likely to establish as an invader. So we have most of the Eastern US and a little bit out West. So this invader is really problematic, not just for us here in Ohio, but throughout the US. So this kind of led me to think about what habitat characteristics make Callery pear more likely to invade. So I published this study in Biological Invasions with our Metro Parks partner, Grace Deach, and it was funded by the Ohio Biological Survey and Ohio Invasive Plants Council. The first thing I wanted to understand is how the land use surrounding these early successional landscapes can influence the invasibility of Callery pear. So a lot of our restoration sites look something like this, where you have the site sandwiched between roads, residential areas, some guy's farm, and then like a forest. So it's really spotty within the landscape. So I wondered, within these environments, what kind of land use around the restoration is going to change Callery pear invasion? Maybe forests surrounding our restorations could lead to woody encroachment because you've got seed available from all of these woody plants on the forest edge that could come through. Callery pear is bird dispersed. So anywhere there's a good bird perch, we might see Callery pear. Something like roadways, which often have lines running on them. And then residential areas are also really popular places for calorie pear to be planted. So if you drive through any of the like Ryan's Homes areas around here, each yard has a calorie pear planted in it, like while the house is being put up. They're just like, here's a house and a calorie pear. So we're seeing lots of calorie pear come from those kinds of areas. What we found is that calorie pear cover was higher closer to forests. So in the site, if we measured the forest edge as 0, which would be 0 here on the graph, we found more calorie pear closer to that forest edge. Now, this makes sense thinking about the dispersal of calorie pear. If it's dispersed by generalist bird species, which also forage on the forest edge, they're going to go eat calorie pear seeds somewhere else, fly over into your site, sit in the trees, eat other stuff, and leave a seed rain behind, which can start an invasional front moving through the site. We did not see any correlation between calorie pear invasion in residential areas and roadways, which is probably because it's really about that dispersal mechanism. We also thought to ourselves, okay, higher species richness could decrease the invasibility of calorie pear. What if we compare some of our invaded sites to our not invaded sites? Will we see that our invaded sites just have lower species richness, and that's why they're being invaded? But what we found is that our sites that were invaded actually had higher species richness than our not invaded sites. And our not invaded sites were typically dominated by grasses. If you want to talk about that later. But thinking about our invaded sites, when we took a look even closer in just our invaded sites, we did see that calorie pear cover here on the y-axis was higher when there was lower grass cover and lower forb cover. So within those invaded prairies, where there was less grasses and forbs taking up space, there was more calorie pear. Now, does this mean that calorie pear established there because there were less grasses and forbs there? Or was calorie pear negatively impacting the plants that were there, leading to a lower cover of our grasses and forbs? That wasn't clear in this study. So my next couple of questions kind of stem out of that. What mechanisms are causing lower forb and grass cover where calorie pear is established? Could it be directly via something like competition or indirectly through alterations to soil chemistry? Let's think about competition first. So many invasive species are allelopathic, so they release secondary metabolites that are really bad for the germination of native plants. So either through the decomposition of their leaf litter, leaching off the leaves, or exudation from roots, they're giving off chemicals that are bad for other plants. So we wanted to know if calorie pear was doing this. So we looked at the germination of some common prairie species when incubated with either water or calorie pear leaf leachate. You can think about the leaf leachate as kind of like a tea. If I take my really dark red calorie pear root leaves and put them in the fridge in water for a couple of days, it comes out in this really dark color. So we're incubating basically calorie pear tea versus water to see, does this tea harbor bad chemicals for native plants? What we found, I'll just show you a couple examples. When we looked at Canada wild rye, we have percent germination on the y-axis over 21 days on the x. We found that when incubated with water, close to 50% of the Canada wild rye seeds germinated. When incubated with leaf leachate in the triangles, about 10% germinated. So it's a pretty reduced germination rate of that species. Indian grass showed a similar pattern when incubated with water in the squares. We had around 30% germination compared to like 15% germination with leaf leachate. Black-eyed Susan showed a very dramatic response to this leaf leachate, where almost 0% of the seeds germinated, but close to 80% germinated with water. And yellow coneflower also had a pretty reduced germination rate, close to 75% germinated with water, and close to 20% germinated with leaf leachate. So I'm pretty confident in saying that calorie pear reduces the germination of at least some native species. It is definitely doing something bad to the plants surrounding it. Okay, but could it also be indirectly altering the native plant species through alterations to soil chemistry? So invasive plant species tend to have really, really fast decay. They have a lot of nitrogen in their leaves, so they break down really quick because everything wants to eat nitrogen, leading to more nitrogen within the soil. So they're changing the the concentrations of carbon to nitrogen within the soil where they are invasive. So we wanted to see if calorie pair was doing something similar. And this was a study published in Applied Soil Ecology with then undergraduate Grace Attia, and it's chapter 3 of my dissertation. We looked at enzyme activities underneath cut and resprouting trees, so little stems of trees, versus large and intact trees, to see how soil chemistry was changing underneath smaller and larger trees, respectively. We measured beta-glucosidase activity, which cycles really labile are easy to break down carbon. It's typically higher when there's more nitrogen, so where there's invasion, we usually see more beta-glucosidase. Peroxidase does kind of the opposite. It breaks down really hard to break down carbon, stuff that nothing wants to eat. It's lower when there's more nitrogen and can be suppressed by invasive species. So we looked at these to see, okay, are these large trees that have tons of leaves changing the soil chemistry enough that we would pick it up in changes to soil enzyme activity? So we would expect that our large untreated trees would have higher beta-glucosidase activity than these smaller trees that don't have as big of a leaf input. And that peroxidase activity would be higher into these smaller trees than these larger trees. We saw the exact opposite, of course. So instead, we see a higher beta-glucosidase underneath these smaller resprouting trees than underneath our larger trees. And this could indicate, oh, and the opposite for peroxidase, sorry. So higher peroxidase, especially under these large untreated trees. It's possible then that calorie pair does have a higher carbon to nitrogen ratio in its leaves than native species, unlike a lot of invasive species that we would typically, or what we would typically expect to see. Although we did not measure that, so that would be a good next step following this project. However, we did find that calorie pair reduced soil pH. So when we looked at soil pH underneath trees from small to large size, underneath our large trees, we had a much lower soil pH. Could this mean that the leaves are acidic? Turns out they are. So when we did the allelopathy study, we were able to find that the calorie pair leaf leachate had a pH of 4.5, which is pretty low. So where you have a huge leaf impulse, a huge leaf input from these large trees, it would make sense that your soil is becoming more acidic underneath those trees. We also saw that our enzyme activity for peroxidase decreased with soil pH here on the X. So it could be that the leaf chemistry is changing the enzyme activities, or that these enzyme activities are responding to changes in soil chemistry. So that wasn't totally clear here either. Now, calorie pair does form a dense monoculture where it invades. And my theory is that it's possible through a soil acidification and allelopathy, it could prevent the establishment of some native species. But if it does have a high carbon to nitrogen ratio, adding carbon to the soil could suppress invasive species. So if it's suppressing native species and invasive species, it would really be lending to its own dominance and its ability to form these dense stands. So we should try to remove it. It's really what it comes down to. It is doing bad things to the environment, quote unquote bad. It's changing the soil in a way that's bad for native plants, and it's interacting with them negatively as well. So my recommendation is that we continue to remove calorie pair, but ensure we're using accurate and well-timed herbicide application in order for it to be successful. Okay, so you've made it to the end of my talk. Thank you for sticking around. I have a couple takeaways just to summarize the findings. So first, the restoration of soil chemistry does occur on a long time scale during restoration in a way that benefits tree seedlings. We do see that soil chemistry and plant communities can shift in a short time scale when they're treated with direct seeding and soil amendments, which could be really exciting for tree seedlings. We see that calorie pair invades on forest edges, regardless of species richness. Calorie pair reduces the germination of native prairie seeds. And finally, calorie pair changes the soil chemistry in a way that it may inhibit native species establishment and promote its own invasion. So that's the end of my talk, but a lot of times people ask, so Michaela, what are you gonna do next now that you're done here? So I was gonna answer that before it was asked. I'm gonna hang out with my kid mostly for the next month and have a really fun holiday season, hopefully. If you haven't seen me in a while and are on Zoom today, I'm also heavily pregnant. So I will be hopefully getting this child out and spending some time at home. I want to say relaxing, but I'm probably not going to be relaxing. And then once life calms back down in a couple months, I'm going to join Dr. Lickstein's lab at the University of Florida as a postdoc. So with that, I'm happy to take any questions. If you're on Zoom, please go ahead and put questions in the chat.
This is for general audience questions.
Yes.
But I maybe actually answer, but like, to what extent do native plants leachate potentially facilitate other plants growing, or is it all an inhibitory effect such that any leachate is going to inhibit growth, or that certain like calorie pair is particularly bad?
Sure. So for example, the question was, does all leaf leachate or these leachates, is it always a negative response for other plants around them? And the answer is it depends on the plant species and it depends on the successional stage. So in a lot of like the early successional like literature, a lot of times the leachates from the previous stage are what inhibit the previous plant community from continuing on and actually then promote the next one. So it can be really complex and it can change based on your soil microorganisms, the soil chemistry. So the argument there is that measuring the specific chemicals and understanding that context is really important to make those claims. Yes, Jen.
How realistic is like do all these soil transplant, right? Is it obviously a greenfield work? How realistic is it to be transplanting soil from established environments into these fallow areas? Like, does that damage the like where you're taking the soil from, or can you transport enough soil for it to like be a feasible wide scale practice?
Sure. So the question is, what's the feasibility of using whole soil like on a large scale? And that's a really good question and something that Metro Parks was definitely like nervous about with me doing that. So I collected 400 gallons of soil for this project from 2 sites. The way that we strategized it was I made like a grid at each site to make sure I was only taking a certain amount every once in a while and I wasn't damaging too much of the property. I don't know though, that's a really great question. It depends on the scale of the restoration and what you want to do and who's in charge and who's comfortable. And like, probably nobody is going to let you go into their relict prairies and take soil or any of their prairies where they know they have rare species. So then the quality of your soil too can change based on what you have access to.
And how much of the seed bank are you moving with that soil?
Yeah, so the seed bank is really interesting. I'm not sure, I don't have a good answer for how much of the seed bank we're taking with us, but I'm really curious about the plant communities in those whole soil areas to see if they're more invaded or what the seed bank did, like what happened. Because we definitely didn't sterilize, we didn't sieve, it was 400 gallons. We just mixed it and sprinkled it on, so. Yeah, Melanie.
This was a terrific presentation. I really enjoyed it. But this might sound like kind of a zany question, but is there like any kind of like soil farming that happens where people like are trying to like propagate these soils just and maybe either to sell them to someone to like restore land or just like to propagate them on a large scale rather than just tweaking small pieces, just like focus on the soil and the microorganisms themselves to kind of like farm the soil to increase microorganisms, mycorrhizae, things like that in the soil.
Sure. So the question is about, can we farm soils in a way that promotes microorganism growth and then use that instead? And it's really interesting. I haven't thought about that in the context of like farming bulk soil, but I do know people you can culture a lot of different microbes. Not a lot. You can culture a small amount of microbes where it's really successful and create a really nice culture collection where you can take that and put it on plants. You can buy like commercially, you can buy mycorrhizae. but they're all really generalists. So it's a really interesting question, because if you could make the exact community you wanted and really design it, you probably would have more success. So it's not a great answer, but it's really interesting.
Like I said, maybe question.
Along those lines, are there more any like chemical solutions like or fertilizer, nitrogen fertilizer, or things like that to kind of do the same thing, except not taking?
Yeah, so the question was, what kind of chemical solutions could there be? So we can think about, like, if we want to add more carbon to the surface soil, it's just like when you're, it becomes about the scale then too. Because if you're thinking about the restoration site that I did, I had to buy like $5,000 worth of mulch to mulch a quarter or half of each of my plots. So it becomes a question again of feasibility, like, can we really buy that much mulch or whatever else would be required? We're kind of trying to overcome the fertilizer use that had happened during all of the agriculture, because my goal would be to dilute the amounts of nitrogen and other things like that to give our native plants more of a chance to survive, instead of the ones that just like cling on to the nitrogen right away. So does that help? Is that what you asked?
So there's really not.
So
Like you put nitrogen in through fertilizer. Like that.
yeah, so adding more carbon to dilute it is like, that's my biggest goal. And so people do all sorts of stuff. So I chose leaf compost because it was locally sourced. It was like a sustainable option that Metro Parks was okay with doing, and that they went through their suppliers. But a lot of people use things like biochar, where you take tons of like corn or something and totally burn it and apply that to the surface. You can do sawdust, manure. Those are like some of the most common things that we see. Yeah. Mhm.
Okay, so I think we'll retire now so that continue. Thank you one more time. As I mentioned before, we'll have refreshments, but not quite. I think we'll get stuff in half an hour.
And thanks for joining everyone on Zoom.